Alrighty, so today's lecture is going to be about enzymes. So again, if you all picked up the little blue book with all the notes in it from the school, make sure you follow along and you can fill them in as you go. So again, today we're going to be talking about enzymes. So this is just a little bit about what we've talked about in the past. So all of life is built on chemical reactions. We talked about this way back in Unit 1 with characteristics of life. And all living things have metabolism and homeostasis. So metabolism, all the chemical reactions in our body, and homeostasis, the ability to maintain a stable internal condition. And all of this is dependent upon enzymes. So again, the metabolism, all the chemical reactions that happen in our body every day. And enzymes assist in this by either forming bonds between two molecules. So an example that we're going to talk about is dehydration synthesis, when you put two things together to make one. And they also help break bonds between two molecules. And an example we're going to talk about is a hydrolysis reaction, and that's when you take one thing and break it apart into two or many. So it's very important that we are able to utilize what we eat to put together to make the nutrients that we need, but also that we can break down what we eat. So first we're going to talk about energy releasing reactions, which is when we digest or break down polymers. And remember, polymers are long chains of any of the macromolecules. This is going to be a hydrolysis reaction because hydrolysis adds water to break a bond. There are also energy absorbing reactions, which require energy to be put in for the reaction to happen. And these are the ones that build polymers. It takes energy to put things together, and you release energy when you break them apart. Now, dehydration synthesis, dehydration means you take away a water to make something. So synthesis, it means to make. So here are the graphs for these two reactions. So for the energy releasing reaction, we can see that energy is only released in the bottom part of this graph in purple. So when our graph starts, our reaction starts, it takes some energy to put in, but ultimately energy is released out of this reaction. Whereas an energy absorbing reaction, it only ever takes energy, it never releases it. It always stays above our purple line, our dotted line here. And so only energy goes in, nothing is released in this type of reaction. So we require energy to live, and this comes from energy releasing reactions. And oftentimes we can couple an energy releasing reaction with one that requires energy to help the reactions happen in our bodies. So when we break a bond and we release energy, that energy can then be used to make another bond somewhere else. So your question might be, if some reactions release energy, they're downhill, then why don't they just happen automatically? Why does it need something to help out? Why do enzymes have to come in and help? And that's because bonds are very stable. Remember, a covalent bond is sharing of electrons. And when they're happy and sharing, they're perfectly stable and they don't want to break apart. So we need something to help them out in that reaction. So breaking down of anything, even though it ultimately releases energy, it requires an initial input of energy to go in before it can be broken apart and release energy. And that input of energy is called activation energy, the energy needed to activate or start the reaction. So the amount of energy needed to start a chemical reaction is the activation energy. And it kind of looks like an energy hill or an energy speed bump. So in our graph here, the distance between the dotted line and the top, or the peak of our graph or our reaction, is called the activation energy, because that's the energy that we need to put in to get over the top of the hill. So I have to put in at least this much to get over the top of the hill. So that is our activation energy. Once you get over it, your reaction just keeps on going and eventually energy will be released. So you might be asking, well, what are enzymes? What the heck are they? So enzymes are special proteins. So we talked about proteins made up of amino acids, and proteins that are enzymes are going to help facilitate chemical reactions, and facilitate just means to help. So they're going to help chemical reactions happen. And they make reactions go faster, so increasing the rate of the reaction, but they're not used up, which means enzymes can help reactions multiple times. They're not used up, they don't disappear, they remain and can help other reactions happen, but they make the reactions go faster. So they speed up the reaction by reducing the activation energy. So essentially what an enzyme is doing to our graph is taking the speed bump from which it was up here, and it's moving it down to here. So we have to put in less energy. Our little speed bump, our activation energy is much, much lower. So enzymes speed up chemical reactions by lowering the activation energy of the reaction. And they're needed for most biological reactions. So most of the reactions that happen in our body require enzymes. And if you look at this graph here, we see the word uncatalyzed and catalyzed. A catalyst is something that speeds up a reaction. So if it has a catalyst, or if it has an enzyme, it's sped up with a lower energy. Uncatalyzed means no enzyme, so the activation energy is higher. Enzymes are also highly specific. So we talked about proteins, we said they fold up into specific shapes. So they work like a lock and a key. Many people have keys, but they all open different locks. So they're very specific and they can only help one specific type of reaction. And it's the shape of that enzyme that's going to make it work. Kind of like the shape of my key is exactly what makes it work in my front door instead of yours. And they have to fit together perfectly. So here in our diagram, this is my substrate, or the item that's going to be acted on. The purple is my enzyme, and as you can see, the two, they fit together perfectly. If there was a different shape here, it wouldn't be able to work. So they have to fit together perfectly in order to work. So a catalyst, again, I mentioned this before, but catalysts are chemical agents that facilitate or help out chemical reactions by reducing activation energy. So they speed up chemical reactions by lowering that speed bump. And they are catalysts. Catalysts get reactions started. So an enzyme is a catalyst, specifically a biological catalyst, which means it works in the body of living things. So enzymes are biological catalysts that work inside of living things to speed up reactions by lowering that activation energy. So they lower that speed bump. So enzymes lower the speed bump and help the reaction go faster. Therefore, they are called biological catalysts. So you want to be familiar with this graph. We'll talk about it here quickly. So lowering the activation energy speeds up the reaction. The way I think about it is, if I'm standing right here at the beginning of the whole reaction, which hill am I going to be able to go up faster? I'm going to be able to go up the little hill the fastest. I can go over it faster. Or if you were in a car, you can go over the little bump the fastest. So the smaller bump goes much faster, and that's going to be your reaction that is catalyzed, which means it has an enzyme. Whereas if the enzyme wasn't there to help out, we have this huge hill we have to get over before the reaction happens. It's called uncatalyzed because there's no catalyst. So again, our enzyme, which is a catalyst, speeds up our chemical reaction, because I can go faster over the little bump, by lowering the activation energy. So kind of think of it like a speed bump. The lower it is, the faster you can go. So here's some vocabulary you'll want to be familiar with. So substrate, the substrate is the reactant that binds to the enzyme, the thing that the enzyme is working on. If I want to take, I don't know, my little toy here and split it into two, this is my substrate, and the thing that comes and helps it break is my enzyme. So my hand is the enzyme, this is the substrate. Substrate is the thing that's going to be acted on, the thing that's going to change. So if you see here, here's my substrate, and eventually it's going to be broken or changed in some way to become a product. The enzyme substrate complex is created when the enzyme and the substrate are fit together and just sitting together. So while they are together, it is the enzyme substrate complex. And it gets its name because the enzyme and the substrate are bound together. That's when it's actually doing its job. The active site is the place on the enzyme where the substrate fits. And this is the part that's going to have the special shape. So on our enzyme here in blue, you can see the little red line, and it has a special shape that fits perfectly with the substrate. This special shape is where the enzyme is active and does a job. So it's called the active site. And then the product is what's made in the end result of the reaction. And as you can see here, our enzyme comes in, helps do the job, makes products, but our enzyme is the same and it's able to do this reaction over again. So enzymes can be used over and over again to do the same type of reaction. They're not used up, they don't disappear, they keep on doing their job. So we start with an enzyme and a substrate, and after the job is done, we have our products. So let's do a quick review. So activation energy. Activation energy is the energy that is needed to start a chemical reaction. We might be asking, how do enzymes actually lower activation energy and speed up the reaction? They do this by helping to create bonds, so to put two things together. Maybe I want my two pieces to become one, so I put them together to make one thing. So I make a bond. Or I break it and pull it apart. So enzymes help by making or breaking bonds. They are very, very specific. They work like a lock and a key. They can only work with one type of substrate, and that substrate has to fit perfectly in its active site. If they don't fit together like a lock and a key, it's not going to work. And they are typically named for the reaction that they help catalyze. So what that means is, let's look at a couple. Typically, the reaction is going to end, or the name of the enzyme is going to end in ASE. So an enzyme that breaks down sucrose is called sucrase. We know it's an enzyme because it ends in ASE and it breaks down sucrose. So it's usually named for what it does and ends in ASE. Protease breaks down proteins. Lipase breaks down lipids. DNA polymerase builds DNA. So they kind of get their name for what they do, and most often they end in the letters ASE. So again, they work together like a lock and a key. You start with your substrate and your enzyme. They fit together like a lock and a key. The enzyme substrate complex is when they're nice and fit together. And when they come apart, we have two products, and the enzyme is left to continue for another day, doing the same job again. That's why we call enzymes reusable, because they can do the same job again. They're not consumed. And as long as they can only do one type of reaction, but they can do that one type of reaction thousands of times. So keep that in mind. They don't disappear. They're not used up. They can do their job over and over again. However, there are a couple things that can make our enzyme break. And if it breaks, it can't work. The first thing is temperature. Temperature can break an enzyme, so can pH. So temperature and pH can break our enzyme. So the reason this is because our enzymes work best in different environments. Wherever they're working, they're going to work best in a certain environment, And if those conditions are altered in any way, they're not going to work as well. So again, the best condition, the optimal condition for our enzyme is where they work best. However, if we change that condition, enzymes can change their shape. When an enzyme changes shape, it's called denaturing. They can no longer do what they're meant to do. So if we change the pH or we change the temperature, the enzyme changes shape. So let's look at this. Here's our enzyme here. It has our special shape. We called this our active site. It has a special shape. It fits perfectly with the substrate. Everything is good. But if we change the temperature, or we change the pH away from the optimal temperature, or the best temperature, the enzyme changes shape. And let's say now it looks like this. These two don't fit together anymore. If they don't fit together anymore, it can't work. So I think of these, it's similar to an egg. An egg is a protein. What happens to an egg when you heat it up, when you cook it? What happens if you were to pour acid on it? Essentially, cooks it. It changes. It goes from a clear-ish color to perfectly white. We've broken it. We've changed it. And once you change it, you can't get it back. You can't uncook your egg. So changes in pH and temperature denature, changes the shape of the enzyme, and it no longer works once you do that, and you can't reverse it. Once it's done, it's done. So here's our quick speed review. So a catalyst is something that speeds up a reaction by lowering the activation energy. An example of a catalyst is an enzyme. Activation energy, the energy needed to activate or start a reaction. It's also known as our speed bump. Enzymes speed up chemical reactions by making or breaking bonds. So enzymes speed up a chemical reaction by making or breaking a bond. And what two things can affect an enzyme's function? That's our temperature and pH. Remember, if we change the temperature, we change the pH away from the optimal or best temperature, our enzyme changes shape, it breaks, and it cannot work anymore.